Okay, it's time. Good morning and good afternoon, everyone. Welcome to the year 2024. I'm Cherry Kubota. I'm a professor at The Ohio State University. Welcome to Indo Ag Science Café. We have been doing 5 years. I'm so surprised, how fast time passed. This is a communication platform and then also a learning platform for the science and technologies that support indoor agriculture, hydroponics, and controlled environment. So this is a collaboration. This is happening through the collaboration between universities, Michigan State University, Ohio State, Purdue University, and University of Arizona, funded by USDA. So thanks to USDA funding this effort. All right, so I just wanted to go through upcoming cafe. We have a great topics coming up today. John Ertle, PP Systems, going to talk about Tipburn. So I will introduce him in a minute. So the next month we have Robert Conrad from Ball Horticultural Company, who is going to talk about seed technology. You may not be familiar with how important this technology is and then how they are supporting us, because most of you are growing from seeds. March, we have international speaker from Japan, talking about volatile organic compounds, how risky this gap of knowledge is. So that which compounds are problematic, which compounds are safe. He's going to share his experience. He's an entrepreneur in Japan, one of the early founders of indoor farming. So I think it's a great talk. Because of the location he is connecting, we have to start 1 hour earlier than usual. So we scheduled 10 a.m. start. So that's the March 5th. And then April 9th, we have another international speaker. He's a scientist in Wageningen University, the Netherlands. He's going to talk about quite unique application of indoor setting. He has a many project, but he's working on what we call speed breeding. So the indoor facility is a breeding platform for seed production. And then he's going to talk about far-red radiation, influence of that, to accelerate the breeding process. So that's very new. It's more science, that topic than, well, I think it's an application connected, but please look forward to those excellent talks coming up. And yeah, so I'm so excited this year. All right, so OptimIA University, basic knowledge of indoor farming. I think it's completed pretty much. So we're going to start advertising. This is educational materials in-house. We might create opportunities using these materials, but right now it's just for you to self-learn for free, watching lectures and downloading materials. If you go to optimiauniversity.org, you can find all the lectures. So something we are proud of about the accomplishment out of this OptimIA group project. Right. Okay. Dr. John Ertle, PP Systems. He's going to talk about Tipburn management through controlled environment for indoor vertical farm lettuce production. John, some of you are already familiar with John moderated this cafe several times last year. And so he's a great moderator, scientist, and then now is a technology R&D person in private sector. So, well, I will let you introduce John, and then so I'm going to stop sharing and have John here and then start his presentation.
All right. So hi, folks. It's great to see many of you again. So as Dr. Kubota mentioned, I'm John Ertle. I was a PhD and then formerly a master's student in Dr. Kubota's lab at Ohio State over the last 5 years. I wrapped up in the summer of last year, and I took a job with PP Systems in Massachusetts. So PP Systems is a company that designs, manufactures, and sells gas exchange systems. like our Cyrus 4 product. And so I'm working with them now, building some in-house research and development and working more directly with the various users of these types of instruments. But I'm going to talk today about what I did during my PhD. So I worked with Tipburn of lettuce. And so I'm going to tell you a little bit about what we did there and walk you through how we go about managing Tipburn in controlled environments for vertical farm lettuce production. So lettuce in vertical farms is likely the most cultivated crop in these types of vertical farming systems. You can see on the left, this is where I got my start, Vigio Gardens in Akron, Ohio, first vertical farm I worked at. And one of the first issues we ran into was Tipburn of lettuce at this startup. And that led me to Dr. Kubota's lab eventually. But to talk more about vertical farms themselves, typically when you are producing lettuce in these types of situations, you have a large variety of selections. So you might have different products, cut leaf or whole head types of lettuce products. They might be different colors, morphologies, taste, pigments. All of these things are able to be kind of leveraged in that marketplace. And so there's the ability to diversify kind of the portfolio of products that you offer as a vertical farm. However, there's a big issue with lettuce Tipburn, and I encountered it here, which led to a lot of headaches. But basically, you may be able to grow an entire full head of lettuce of high quality, but once you start peeling back some of these outer leaves, you might find this necrotic tissue forming towards the center of that lettuce head, which is a large problem for these growers, as they either have to manually remove this Tipburn or they'll have to throw out significant amounts of product, which is the last thing that anybody wants to do in that situation. So Tipburn itself at a physiological level is really a breakdown of pectin, which is a structural component of the cell wall in these tissues. And so these polysaccharide chains are bound by a calcium cation to keep them stable as they develop and to allow for that leaf expansion to occur without any types of breakages in the cell wall that leads to leakage and eventually necrosis. And so this Tipburn is actually caused and determined by the growth of that lettuce. So it's really a balance of calcium supply and demand. So the calcium supply is driven by this transpiration and the root pressure as these hydroponically grown lettuce take up calcium cations and water in the root zone, and then transport this calcium via the xylem through the plant and out to the different leaves. You can see on the right, the orange arrows kind of indicate the amount of water and calcium that are being transported. The outer leaves are going to receive more calcium because there's more transpiration pulling that water and that calcium there, whereas the inner leaves usually are a little bit shrouded by those outer leaves, blocking that direct movement of water from the inside of the plant to the outside of the plant because of the density of the head. Now, the calcium demand is primarily determined by the growth rate. As the leaves expand and the plant grows, that demand for calcium is going to increase over time pretty linearly with that actual increase in the growth rate. So if we look at this kind of figure, this is typically how a lettuce fresh weight looks like at various time points across an indoor farm. So early on, we have very slow growth, which begins to ramp up and become exponential as you approach the harvest phase. So about 20 to 28 days after transplanting into these hydroponic systems is where we usually encounter an imbalance where the calcium demand, the growth rate, outpaces the ability of the plant to supply calcium to those expanding tissues, leading to the breakage of the pectin chains and then the necrotic tissue formation. So calcium uptake is, like I mentioned, transported via the xylem from the root zone, and this uptake is driven by the mass flow throughout the plant. So during the daytime, this is primarily transpiration driven. So as water exits through the leaves, through the stomates of the leaves, more water is pulled through the xylem, and then more water and nutrients are taken up at the root zone to support the plant's growth. At night, this mass flow is driven by root pressure. As the stomates close, that xylem pressure increases inside the plant and kind of forces the water and the whatever nutrients that are in with the water transported to the leaves. Calcium is an immobile nutrient, meaning it can't be translocated. So although these outer leaves receive a lot more calcium than the inner leaves, it can't be taken and then moved somewhere else in the plant. So the plant has to rely on the supply of calcium at all times to support its expanding leaves and growth, which is primarily why we see this deficiency appear in the young leaves, as the young leaves are often blocked by the outer leaves, so there's not as much transpiration there, and we can't pull calcium from other parts of the plant to support that growth. So typically, we always see tip burn occur in the young leaves of the plant or the meristem itself. So plant transpiration itself can be modified by a number of different environmental factors. For example, airspeed and wind. As more air moves over the leaves, the more water is wicked away from the leaf's surface, and the more transpiration happens as a result. Something like humidity or VPD. Basically, the more water that's in the air results in less transpiration from the leaf, as the gradient from the leaf to the surrounding air, the water gradient, is rather low. When there's a large difference in the gradient, a lot more transpiration occurs as that water is moving from an area of high concentration inside the leaf to low concentration outside of the leaf. The air and leaf temperature also can increase transpiration. The higher the temperature is in that environment, and the higher the leaf temperature is, the more transpiration is going to occur to promote that evaporative cooling and heat loss through the leaf. And also radiation. So whatever the incident radiation is will drive that transpiration. However, in these indoor settings, we're usually using electric lighting, which has a much narrower wavelength sort of range than direct sunlight. So a lot less transpiration is driven by electric lighting, even if the same light intensity is achieved. So in this case, here are 3 examples of lettuce grown in Ohio itself. So on the left is an open field cultivation, in the center, greenhouse cultivation of lettuce, and on the right is the indoor farm cultivation of lettuce. And so I kind of made these icons relative to the amount of transpirational flux they might drive. So outside, we have on the left in the open field, there's a lot of abiotic stress. You have wind going from high speed to low speed and changing over time. Temperature can vary quite considerably, and the sunlight and the incident radiation can change quite a lot depending on the day and the weather occurring in that area. In a greenhouse, we kind of protect against this a little bit. We might have extra lights inside the greenhouse to drive further photosynthesis, but the wind speed and the temperature range are quite reduced because there is not as much, because the actual glasshouse interferes with the environment and protects those plants from some of those conditions. In an indoor farm, we're in a very tightly controlled space. So the amount of airflow, the temperature range, and the light intensity are all very finely controlled and tuned to try and promote growth, but that also may have some other implications. So relatively, the transpirational flux in any of these 3 environments is the highest in the open field and the lowest in the indoor farm. Now, because transpiration is the primary way we move calcium within the plant, then we increase our relative tip burn risk as we go left to right. So really, it's the reason why we see tip burn so predominantly in the indoor farm setting is because of this reduced transpirational flux inside that environment where we see highest tip burn risk. Now, once tipburn occurs on a plant, it can't be rescued. So if we see on the right, there are a number of necrotic areas where tipburn is occurring on these two plants, but there's no way to reverse this symptomology. Typically, once tipburn occurs, unless you intervene in some way, every new leaf thereafter will also develop tipburn. So growers have usually kind of a tool belt of ways to try and prevent this problem from happening in their farms, but there are some limitations to many of these. So the first prevention mechanism is selecting cultivars that are going to be highly resistant to tipburn. However, our limitation here is more of a knowledge gap in how we currently test for tipburn susceptibility. So there are hundreds of commercial cultivars available to growers. These seed companies often do not often publish whether or not they're tipburn resistant, but they're usually testing this tipburn resistance in a greenhouse setting. So we're moving a little bit away from that indoor farm specific type of tipburn sensitivity, and we're looking at greenhouse lettuce tipburn sensitivity in that case. So this is not super useful for the indoor farmers who are not sure exactly what cultivars to select to have a low tipburn risk. Now, another issue could be two other ways we can prevent tipburn are with different types of water management. So during the daytime, we can enhance the transpiration rate, and a number of facilities do this with downward vertical fans or other mechanisms. However, that's sometimes limited by your facility limitations. So this is kind of an industrial building that's more of like a facility limitation icon for this purpose. And because of that indoor facility, you may not want to drive too much transpiration given your HVAC load and other issues. So there may be some issues with water management. At nighttime, you can promote guttation, which is a well-known method to prevent tipburn. However, that means you have to elevate the humidity to greater than 95% for most likely the whole nighttime. Managing that amount of humidity is very expensive, and over time, collection of water on electrical systems and other things could be an issue. Another option is to simply avoid tipburn entirely by harvesting the plant early. However, that might lead to some kind of profit reduction as you are missing out on the fastest growing period of lettuce in those indoor farms. Another option may be to simply slow the plant growth and have a longer cultivation period. But then you may have less harvest per year, and that might also affect your bottom line and could be a major limitation for a grower. Another method is to spray foliar calcium. So by applying an exogenous supply of calcium to the plant, by applying it on the leaves, this can actually prevent or reduce the tipburn symptomology. However, spraying foliar sprays and calcium in a facility might not be the best idea in an indoor facility due to the damage to equipment as mentioned and humidity management of the air. And I did mention downward airflow. However, I wanted to bring it up again because really this is one of the most effective methods we know to reduce tipburn. However, with the vertically integrated structure of many vertical farms, it is very challenging to provide downward airflow of high enough speed to prevent tipburn from occurring in lettuce. So these are kind of the tools in the tool belt of the grower. However, each of them have some number of limitations. And so what we wanted to do was to work a little bit within this to create more resources for those growers. So we ended up settling on about I'm going to talk about three specific research challenges. So the first one is cultivar specific lettuce tipburn risk for indoor farms. So while reported by seed companies under greenhouse conditions, tipburn risk might not be quite accurate for an indoor farm setting versus a greenhouse setting. And then growers themselves are lacking that information to make that their appropriate cultivar selections for their indoor farms. Another challenge was to evaluate a known method to prevent tipburn, slowing the growth, which we generally avoid due to revenue loss, as I mentioned, having a longer cultivation cycle potentially, but at least revenue loss due to less cycles per year. However, tipburn prevention may be more economically feasible than the labor cost of removing tipburn leaves. So it could be possible that in a scenario that slowing the growth might actually be beneficial to your bottom line, but we wanted to look into that more closely. And then also we wanted to evaluate perhaps a new method for growers to prevent tipburn, adding another tool to that tool belt. And our goal was to increase that calcium transport to those susceptible leaves through some alternative mechanisms. So I'll talk about it a little bit later, but we used a mechanistic application of blue light to try and increase this transpiration. So I'm going to start with the first bullet point at the top today, or I'm sorry, I'm going to tell you the hypotheses first before I get into the experiment one. So we wanted to standardize this. We wanted to standardize tipburn testing, or at least provide an example of how you might test for tipburn sensitivity in an indoor farm specifically. So our first hypothesis was that we could develop a tipburn inducing condition for indoor farm risk evaluation. The second was that kind of closed head varieties like romaine have been known anecdotally to have a higher tipburn susceptibility than other lettuce types with a more open structure or morphology. So our hypothesis here was that these closed head varieties would likely have more tipburn than those other butterhead or leaf type lettuce open morphologies. And then also that the seed company cultivar recommendations that are published might not be super effective at predicting the tipburn risk in the indoor farm setting. In experiment two, we looked at end of production lighting to prevent tipburn. So we reduce in experiment two, I'll talk about, we reduce light intensity during the last few days of growth so that we can reduce the tipburn severity, even though this will likely also reduce the yield. Potentially, this strategy could be profitable if the reduction in tipburn is greater than the amount of biomass lost due to that reduction in light intensity. And that this might be a very beneficial mechanism if you're sure you want to grow a higher tipburn risk cultivar. In the third experiment, I'll talk about later, we used dim nighttime lighting to try and increase the transpiration and movement of calcium within the plant to prevent tipburn. So the idea here was that dim nighttime lighting can increase that transpiration and enhance the flow of calcium, but the nighttime, and specifically nighttime blue light plus some red light will likely increase the transpiration the most, and that nighttime light will reduce tipburn severity as we move more calcium throughout the plant due to that transpiration. So we're going to get into kind of the weeds with experiment one here in a moment, but just a quick methods sort of part. So for all three experiments, we did nearly identical types of experimental designs. So seedling growth was conducted in a growth chamber for 2 weeks, then transplanted into NFT system in two separate walk-in growth chambers, identically designed. And within each growth chamber were 4 carts, each serving as our growing area for these plants. And so we also use the carts as a blocking mechanism. But that crop production was about 4 weeks to full size and harvest of these lettuce. We measure tipburn in a few different ways. Right now, seed companies will say things like this is strong against tipburn or resistant to tipburn, but the literature varies widely and how we report tipburn, different indices and other metrics. So we settled on 3 kind of taken from plant pathology. So tipburn incidence, meaning the percentage of plants of the whole crop having tipburn symptoms, tipburn severity being on an individual plant, the percent of leaves that have tipburn symptoms, and then tipburn emergence, which is the amount of time from transplant to the first tip burn sighting on an individual plant. So these were the kind of metrics we used to do our tip burn related analyses. We also took many other measurements, including the fresh and dry mass of roots and shoots, leaf counts, mineral composition analysis. We separated inner and outer leaves to do this mineral analysis, so we could see the amount of calcium in the innermost leaves and outermost leaves. And we also evaluated the plants in their gas exchange twice during each repetition with the Cyrus-3. So in that experiment one, we wanted to grow these commercial cultivars under a tip burn inducing condition to identify their relative risk in that indoor farm setting. Then determine some differences in that severity between the different morphologies of lettuce, and then evaluate the severity against the recommended production systems proposed by the seed company, which could be indoor farms or greenhouses as their recommended system. So we selected a number of tip burn inducing conditions. So elevated CO2, high temperature, high daytime humidity, low nighttime humidity, pretty high daily light integral of 16.7 moles per day. And the goal here was to create as fast of a growing environment as we could. So really try and push these lettuce to grow as fast as possible so that we reach that deficiency point, that calcium deficiency point. And then we also tried to restrict transpiration with the high daytime, low nighttime humidity and a very, very slow horizontal and vertical airflow rate. So only 0.07 meters per second on average in vertical and horizontal directions. And so this was really to try and restrict that calcium transportation through transpiration and ensure that we see this deficiency. So this is about as likely to get tip burn as we could get this designed. We selected 10 commercial cultivars. Now these cultivars were a mix of red and green lettuce types with differing morphologies. And then the recommended production systems by the seed company were either for outdoor cultivation, indoor meaning greenhouse cultivation, or both indoor and outdoor cultivation. So quite an array of different plants here to look at the same time. And to get right into results, after 4 repetitions of this, we have on the x-axis for these coming up graphs will all be the cultivar, and the y-axis is the number of days after transplant in this case. So we see very similar tip burn emergence. So somewhere between, the average was 22 plus or minus 2.5 days. So typically right around that 3-week mark is where we start to see that tip burn emerge in these plants. And then interestingly, we wanted to look at the tip burn emergence and the leaf calcium concentrations at the time of harvest. So when we look at the tip burn emergence on the left side and the outer leaf calcium concentration, there's a pretty linear relationship. But when we don't typically see tip burn on the outer leaves, we typically see on the inner leaves. And there's a much tighter relationship here, although the standard deviation for the number of both the tip burn emergence and the inner leaf calcium concentration are pretty wide range. But we did see that the inner leaf calcium concentration is about 1/3 of those outer leaves. And then the more inner leaf calcium there is, the later you would see tip burn emerge in these plants. And so cultivar to cultivar, that can vary quite a bit as to how much calcium is at least going to delay your tip burn emergence. When we look at tip burn severity, there's quite a wide range as well among these 10 cultivars. So the average was about 40 plus or minus 18% of leaves having tip burn symptomology at the time of harvest. And there are some plants like this Ruche cultivar down here that has very little tip burn compared to something like Klee, which has quite a lot of tip burn, more than 50 up to greater than 50% of these leaves at times. And so this tip burn severity can vary from cultivar to cultivar quite extensively. Now, anecdotally, we often hear that, I've heard at least, that lettuce fresh weight is the best indicator of tip burn risk. So basically, the larger those cultivars get, the more tip burn risk there will be, as the difference between the calcium demand to grow that large of a fresh mass may not be able to be reached with the calcium supply of that size plant. But we found that for each cultivar, there was no relationship between that shoot fresh weight and the tip burn severity. So it seems apparently that fresh mass is actually not a good indicator of the tip burn risk. However, within each cultivar, you can predict a bit of that risk. So within a cultivar, the higher the fresh weight is, the more likely the higher tip burn severity you're going to see. So higher percent of leaves within a cultivar, but between cultivars, it almost seems more like that cultivar's ability to supply calcium to the inner leaves, even in a tip burn inducing condition, is going to be the best indicator of whether or not that plant is going to have a higher low or susceptibility to tip burn. We didn't find any link between the tip burn severity and the lettuce morphological type. So although romaine is anecdotally known as the most susceptible to tip burn, it actually had no difference with any of these butterhead, leafy, or romaine categories in our experiments. And neither did lettuce color. So red lettuce is known to often grow a little bit more slowly than green type lettuce because of the anthocyanin pigments and how that works in the plant. But we didn't see any difference in tip burn severity for that lettuce morphology or color. We did see, however, a small difference with the recommended production system provided by the commercial seed company. So if a cultivar was recommended for indoor cultivation only and not for outdoor cultivation and not for indoor and outdoor cultivation, just indoor cultivation, there's actually a lower tip burn severity for those lettuce cultivars only recommended for indoor production. So seed companies are making more and more selections towards finding tip burn resistant lettuce cultivars, and it seems they are successfully at least recommending some of those cultivars for commercial use. We also conducted a follow-up trial where we tested 17 additional cultivars in this tip burn inducing condition. Out of the 20 total cultivars we tried, we found 5 that had a tip burn severity of less than 10% in this indoor condition. So that's a pretty good sign that there are absolutely some commercial varieties available right now that have very, very low tip burn susceptibility. However, in addition to that, we again found that there was no correlation of the tip burn risk or severity with the lettuce morphology, color, or yield. So it's kind of a case of don't judge a book by its cover with the different lettuce cultivars. Looking at them is not enough to predict their tip burn risk. Rather, it's the kind of the genetics of each of those cultivars that seems to be more closely correlated to and their ability to transport calcium that seems more closely linked to their tip burn susceptibility. So in the second experiment, we looked at end of production lighting to prevent tip burn. So we wanted to reduce the daily light integral to slow the growth and reduce the calcium demand during the period of highest demand, the most rapid growth phase. And we wanted to quantify the reduction in tip burn and potential marketable yield that you might gain from avoiding tip burn in that scenario. And whether or not that's going to provide a positive economic return versus having no reduced DLI and having a lot of tip burn. So bringing this figure back, as that calcium demand increases, we chose to try and decrease that demand only during that last period in the box here by reducing that DLI. So in this experiment, we grew two cultivars that we had previously used. Clee is a very highly susceptible variety to tipburn, and Rex is moderately susceptible. So in this case, we grew the plants for 2 weeks. Then during the crop production phase, we grew at the same DLI up until 16 days after transplant. At that point, we are coming up on that 20 day or so after transplant where we start to see that tipburn risk. So we picked a few days before to reduce the DLI down to 85, 70, or 50% of the total light intensity. I think that was 100% was 17 moles a day or so. And so reducing that DLI by quite a bit for each of these 3 treatments and our 100% light control. So what we see here is on the x-axis, this is our DLI reduction. So our treatments were 55, 70, 85, and 100. And then we have our head fresh mass on the y-axis for Clee and Rex. Now, as we reduce that DLI, we do get a linear decrease in the yield for both of these plants, which is not surprising, and we expected to see that. However, when we look at the tipburn severity, we see a somewhat interesting relationship. For Clee, the very highly sensitive cultivar to tipburn, there was a nonlinear response. For only the lowest light treatment did we see a reduction in tipburn. While for Rex, it was a little bit closer to linear, could be nonlinear, but pretty immediately. So for an 85% light treatment, for example, big reduction in tipburn, similar to the 70% lighting treatment, and no tipburn at all in the 55% lighting treatment. So by just reducing that DLI during the last 12 days of growth, we can see a huge reduction in tipburn or elimination of tipburn depending on the cultivar used. Although this does come with a yield penalty, as we see on the left. Economically, the loss of product or the loss of the growth we missed out on for these lettuce cultivars can't outweigh the amount of tipburn we avoided for this to be profitable. So while we reduced growth up to 22 or 26% for these 2 cultivars, we could reduce the tipburn severity by up to 75 or 100% than the control treatment. So for Clee, so sorry, when we're looking on this figure here, the lighting treatment is our x-axis, and then the y-axis is the contribution to our profit margin. So the white boxes are the marketable value that we're either losing out on or gaining, and then the electrical savings are always going to be a gain as we reduce that electrical cost from running the lights at a higher intensity. And when we see for these 2 cultivars is that for Clee, in that lowest 55% lighting treatment, we can actually get a positive contribution to our profit margin. So in fact, reducing that DLI, saving on some of the electrical cost, and not having as much tipburn material means that we can actually profit an additional $2.55 per meter square of cultivation of Clee. However, for Rex, the loss in growth, that 26% loss in growth at 55% lighting treatment was so large that we actually would be losing close to $15 per meter square in one harvest. So it appears that for highly sensitive cultivars to tipburn, this might be a way to continue growing those specific cultivars with it being economically still viable in the facility. So we're going to move on to our 3rd experiment, which was this nighttime dim lighting experiment. This is a little bit different than the ones we did previously, but we essentially want to increase the mass flow of calcium when there's little calcium demand by applying a small amount of light at night. That is going to try and open the stomata of the plants and increase that transpiration rate. And see if this strategy might be able to help mitigate our tipburn risk. So blue light has been reported and observed to activate phototropin in the guard cells that causes the swelling of stomata. This leads to the stomatal openings that it can exchange gas freely with the air. I'm not going to take you through the whole mechanism, but basically blue light is a phototropin mediated response that can open the stomates and at least assist the alternative photosynthetic pathway in opening the stomata. But it's found that this phototropin mediated response can happen with or without the addition of any other light. So just a small amount of blue light can be used to increase stomatal opening in many species. And it's also reported to be enhanced considerably by the presence of red light. So we designed this experiment with the same two cultivars, Clee and Rex, using our Tipburn inducing condition, but we added in this nighttime dim lighting component for the 8-hour nighttime. And during the night specifically, because we don't want to contribute to calcium demand due to growth. So when we apply this blue light at night, the light transitions from a daytime spectrum to a nighttime spectrum, and it makes your computer screen look a little bit broken due to the overexposure, but that's what that transition looks like from day to night. And we used two different spectra during the nighttime. So we used either 100% blue, which I'll call B100, or we used 80% blue and 20% red light at night. And we selected an intensity of about 30 micromoles per meter squared per second. So on a standard, on kind of typical light response curve, where photosynthetic rates on the y-axis and our light intensity PPFD is on the x-axis, above this light compensation point we see here, we're going to increase calcium demand. Now in this case, the light compensation point is right around 85 micromoles per meter squared per second. So that's why we selected this 30 micromole target to fall well below our light compensation point somewhere in here, which we did test before running this experiment. So I do have a more exact number of where the compensation point is. But this was well under our reported light compensation. So just to kind of preface some of the results, we didn't see any change in yield adding this small amount of nighttime lighting. So we had our control and then blue light at night and then blue and red light at night. It contributed less than a mole per day. So no change in yield is good. That means we were below that light compensation point. And the daytime gas exchange was actually quite similar between all these treatments, regardless of the cultivar. So no real daytime effects from this nighttime treatment. But we did see some differences in gas exchange when we evaluated the lettuce at night. So on the y-axis here, we'll see our stomatal conductance in millimoles per meter squared per second. And then we have our treatment. So either our blue and red light, or control nighttime darkness treatment for Clee and Rex. So for Rex, we see under blue light, a big increase in stomatal conductance at night over the control, and more so than the blue-red combo did. Clee has a very similar trend, but these were not significantly different statistically, so we can't quite say the stomatal effect was profound there. But we do see a big difference in the transpiration rate at night for Clee and Rex. Under blue light, we do see quite a large increase in transpiration. Sometimes I think it was 40 to 50, about 30 to 40% or so increase in transpiration rate during that entire nighttime. So we thought, this is great. Okay, we might actually be driving more calcium through the plant that might solve our tip burn issue. However, when we look at tip burn severity at the time of harvest, all of the treatments performed about the same. There's actually really no difference between in tip burn severity, incidence, emergence between any of these three lighting treatments. So we thought we should rerun this experiment with some modifications. So we did a follow-up experiment. We only focused on the 100% blue light. We also used our nighttime darkness as a control, but we also added fans to each of these treatments in another group. So B100 light was replicated twice, but one of them involved the use of fans, taking that 0.07 meters per second average vertical and horizontal wind speed and changing it to 0.4 meters per second, which is just about the tip burn prevention recommendation for lettuce and indoor farms. So we used this B100 light and we used nighttime darkness like our control plus this downward fanning. And what we saw here was for Rex during the nighttime, very similar stomatal conductance across all of these treatments. So surprisingly, no big differences between the B100 and the control. And you can see that these underscore F here is marking that this was a fanned treatment as well. For CLE, however, the stomatal conductance for the fanned treatments was much, much lower than that for the 100% blue nighttime lighting or the control treatment. And that resulted in a reduction in tip burn severity in both CLE and Rex. So a little bit counterintuitively, for both of these cultivars, the presence of fans essentially eliminated tip burn, except for one leaf of one plant in this B100F treatment for CLE. So basically, this downward fanning was highly effective, and the B100 versus control treatments were exactly the same, really, in just about every way we can measure them. No real difference in tip burn severity or gas exchange in those two. And we can see here that for many of these plants with the no fan treatments, we can see tip burn popping up for just about every plant, whereas there's no tip burn on the other side of the screen. So basically, nighttime lighting increased this whole plant transpiration rate, but didn't prevent tip burn at all. And we were kind of wondering what this was about for a while, but at this point, we think that this is really just a case of transpiration versus guttation. So by supplying this blue light, we ended up increasing the whole plant transpiration. Now, the calcium in the water are only going to flow to places where the transpiration rate is highest. So if we're increasing whole plant transpiration, we're not specifically targeting these inner leaves in the meristem that have the calcium deficiency. So it wasn't actually effectively changing the amount of tip burn that we would see in the plant. Whereas nighttime humidity treatments that have been published many times usually cover the plants or increase the humidity of the surrounding air by so much that the plant actually is not transpiring much at all, and it forces that xylem water and nutrients to flow to the tips of these leaves. And in this plant, you can see the water droplets forming and coming out of the hydrothode on the edges of these leaves. So while we may have increased the whole plant transpiration, we're not really successfully driving enough calcium to those areas to be effective, which is why guttation might have been a more effective mechanism to try and look into in that case. But just kind of conclude what we found here. During that tip burn testing trial, we were able to create a tip burn inducing condition. We found that the tip burn risk of lettuce is relative to each cultivar, and that morphology and color are not super indicative of whether or not you're going to have tip burn risk. And seed companies are improving in the recommendation of cultivars with lower tip burn risk, and it seems that there are many on the market now that could have potential in being low tip burn risk cultivars. From the end of production lighting treatments, we found that reducing the DLI during the last few days of before harvest, last 12 days, can reduce the tip burn severity. However, you do suffer a known yield penalty. So this might be somewhere where this can be optimized a bit further to a cultivar specific level to determine whether or not this is an appropriate economic approach for different growers. It can increase marketable value on a square meter basis, which is really the driving force to maybe try out this tool. However, it is most appropriate for the highly tip burn sensitive cultivars, which there are many. So it may be prudent to try and use that type of mechanism. From the nighttime lighting, we found that the dim blue light can increase the transpiration of lettuce. However, this doesn't relieve any tip burn risk. And instead, we found that downward airflow is highly, highly effective at alleviating tip burn, and is one of the most often given recommendations we have for preventing lettuce tip burn. So I just want to say thank you to everybody, and you may have seen my research project and Dr. Kubota on the if you've watched an Ohio State football game in the last 2 years. We've been showing up somewhere about every halftime for about 1 second or so during these games. So I grabbed a screen grab for fun. But thank you for listening to my talk, and I'm happy to take any questions.
All right. Thank you so much. I really appreciate